Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Sven Heinz from UCSD on this show. Please let me briefly introduce you to our audience. You got your PhD from the University of Regensburg in 2002. You then moved on to do a postdoc at UC San Diego in 2004 and became assistant project scientist in 2008. Then you moved to the Salk Institute as director of next generation sequencing. In 2016, you then moved back to UC San Diego as assistant professor and became associate professor last year in 2023. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
So actually I started as a chemist, and I guess, how did I become interested in biology? I really became interested in the question, how do you fix the sun energy? And so I got into photosynthesis, and then I thought I was going to be, from physical chemistry, spectroscopy, combine that with biochemistry and stuff. And, but then I realized that wasn't really feasible at the time. This was in the 90s. And so I got into biochemistry through that, though. And then I did a lab course at the university, and in Regensburg, actually, that was 3-week intensive, like morning to evening.
Yeah.
And that was really fun. And then somehow at the cheese counter in a supermarket, a colleague of mine was like, I was going to be doing physical chemistry, spectroscopy and stuff. She was like, we have this thesis open and it's biochemistry in the hematopoietic system and protein purification. And I was like, nah, I'm going to build a time-of-flight mass spectrometer. I know what I'm going to do. But then somehow I ended up going there anyway. It's been that ever since somehow.
So you must be very interested in the artificial photosynthesis things that are going on currently.
Yeah, well, I've kind of lost track. I was basically back then. I was like, oh, could you do this in solid state? Like in inorganic chemistry, basically.
Okay.
But as usual, I would get caught up in the smaller questions, like how do you even get the water to the reaction surface, and stuff like that. And then I was just like, you know what? But no, I got to look that up. I'm actually not up on this at all. I'm just stuck in grand writing mode and not really reading much what's going on.
Yeah, we'll come to that a little bit later, but let's focus on what you're doing or what you have done since this encounter at the cheese. Cheese. So your science centers around developing high-throughput sequencing and biochemical techniques to further define the principles governing gene regulation and chromatin organization. And you also work on making chromatin profiling methods suitable for clinical diagnostics use. I want to start, as we always do, in the beginning. In this case, I want to start when you were with Christopher Glass, however you pronounce this. There you demonstrated in macrophages and B cells that collaborative interactions of the common factor Pu1 with small sets of macrophage or B cell lineage determining transcription factors establish cell-specific binding sites. Which is always interesting to see what transcription factors really do when it goes to determine lineages. So can you talk about the study and what you found about the system of layered regulation of transcription factors?
Yes. Yeah, so that was really cool. And it also started with a complete fail. So I joined the Glass Lab because the Glass Lab was part of a consortium here at UCSD together with Jeff Rosenfeld and Bing Ren and Shadlong Fu to make microarrays where you had all the promoters of this is shortly or basically during the human genome coming out. All the promoters spotted on a microarray, and then you could do chip on chip. So you do chromatin immunoprecipitation for like a transcription factor and see what was bound to that factor, or in other words, where was it bound in the genome by hybridizing that DNA to those arrays. And basically it was like a technology, but then it was a question, what do you do with it now that we can work it, and the first thing, of course, was epigenetic marks and stuff. And then, but I was always interested in, how do transcription factors actually do their thing? And there was this one question that this factor Q1 is important in early hematopoietic development. And it's, when you looked at the literature, there were genes, and so it's important in B cells for B cell and macrophage development. Those are two different lineages in the hematopoietic tree. And when you looked at the papers, it was like, oh, Q1 is really important for this B cell specific gene. And then it was like, oh, and Q1 is really important for this macrophage specific gene. But that's not expressed in B cells. So how does this work? And so then the idea was like, hey, let's just, isolate macrophages and B cells from, in this case, from mice. And actually we also did neutrophils, but anyway, use this to pull down Q1 and see where is it bound in these different cell types, because they have the same genome, right? So it should be binding to, either it binds to the same sites or there's something about the sites that it binds to in each one that is different that tells us how this works. And so we did that and hybridized it to our promoter microarrays. And it looked like there was no difference. And then at first we thought the arrays were broken and like maybe they don't work. And so we did the same thing with a commercial vendor and got the same result. And it was like, there's really no difference. And this doesn't really jive with the publications. And how does this even work? And that year, 2007, UCSD bought the first Solexa sequencer, which is now Illumina. and we had the chance to put our stuff on there, and that also horribly failed because nobody knew how to run the machine really. And but it was so promising that another one was bought, and then I was actually trained on that, and we ran that same sample that we had run on the microarrays like half a year earlier on that sequencer, and it turned out that the same factor in the same genome, they were interacting in different places in different cells, and those places were all not at promoters. That it explained why our promoter arrays totally failed. I mean, they worked, they didn't fail, but it was the wrong idea to start out with, And then my colleague, Chris Benner, who was a bioinformaticist, he had written a really great software to do motif analysis. And so he threw the data, and he had been kind of following the Chipstick thing coming up that also came out in 2007, basically sequencing the chromatin IP product. And then his motif software found that the top motif was what we expected, the P1 motif. But if you looked at the cell type specific motifs that also came down with this, that were enriched at the sites where P1 was bound, you got the motifs for factors that were only expressed in that respective cell type where P1 would bind. And so then the next step was like so until then, at least for us, the idea was, okay, if, sure, you have a factor that binds DNA, but that's a really short binding site, and that's really not enough to even distinguish between the sites that are in the genome and that are actually bound, because there's like 10 to 100 times more sites in the genome that actually are bound, and that wasn't really understood either. And so the idea was always that you get a ternary complex. You have this factor, like let's say P1, and then this interferon response factor 8 that is known to interact with P1, and together they make a bigger binding site. So make a ternary complex between the DNA and the two factors that have to interact with each other. But when we looked at where those motifs were, so for IRF8, they actually checked out the motif was right next to the P1 motifs, was like this like line distribution, 2 lines, where the motifs are across all the binding sites. But if you did this for any of the other transcription factors that were specific to each of the cell types, you would find like a bell-shaped curve of distribution, which didn't make sense, because if you think about DNA, right, 10 base pair spacing of the helix, if you bind to each other, cool, then you're right next to each other. But then you you turn it away, and the distance increase, you can't really variably change that. And so you can't really interact anymore. Your factors are like 5 microns, so 5 nanometers diameter. And, each turn is 3.4. So it's like you turn this one turn away and you can't touch each other anymore. So that ternary system didn't work anymore. And then the other thing was that in a different project in the glass lab, we found that, when you activated another transcription factor that wasn't per se bound to the genome, it would prefer to go to places that were already bound by P1, and these potentially other factors. And anyway, we kind of dove into that more, and we found that, yeah, it's like these factors, they help each other bind. Like if you only have one and you have weak binding sites for both, that's not enough to stay bound to the DNA because you still have nucleosomes floating around, right? I think that's really the idea that the Brownian motion or the active motion through chromatin remodelers of nucleosomes kind of kicks off transcription factors, right? Because they have to compete with nucleosomes, 15 million of them per genome to bind. And so if you have two, you're kinetically and like probably more like a mass effect wise, you're favored to be more bound than not bound and displace those nucleosomes. And then the next thing is once you have multiple factors bound, then you can recruit like co-regulators that help, remodel the nucleosomes and stuff. So that was like a really long story.
So the places you found those binding sites were then probably enhancers, right?
Yeah, that was actually, because they were not at promoters. And actually in the lab meeting, somebody asked, so where do you think this is? And I was like, enhancers. And then I think there must be enhancers because it's not promoters. And somebody else was like, nobody believes in enhancers anymore. It's 2008. It's funny. And it turned out, yes, they are enhancers. They're marked by enhancer marks. They function as enhancers. Yeah.
It's like, what about the internet? Nobody believes in the internet anymore.
Right. Yeah, exactly. That doesn't exist.
So your next paper was then published in Nature. There you investigate the genome-wide effects of sequence variation on lineage determining and signal-specific transcription factor binding, epigenomics, and transcriptional outcomes. So a very broad kind of study. So what did you find there? Was this a follow-up to
that was a follow-up, yeah, because we were like, so what we had done in the previous paper was to use inducible systems to take knockout cells that were deficient in one of those factors in the mix and basically reintroduce them. And then you could see that, if you introduce that factor, then you get nearby binding of the other factor that wasn't bound there without the inducible one. But there was kind of, we didn't really have enough systems to test this with all the combinations that we want. Plus, you have transcription factor redundancies. So P1 is an exception in a way. I mean, even though it's part of a big family of ETS factors, most of the other factors like CBP, AP1, they form large families that are all expressed in those cells. So if you just get rid of one of those factors, there's still redundancies built into the system, right? But we figured that what if you change the sequence? If you look at the genetic variation between individuals, or in this case, mouse strains, it doesn't really matter what the redundancy on the factor side is if you change the motif, right? Yeah. So basically we did kind of a similar study, but then compared the macrophages from two different mouse strains. And basically that corroborated our initial findings from the first paper that, you could. So if you have two motifs for different factors that bind in the vicinity of each other, if one variant hits one of those motifs, then both don't bind anymore, right? And so that was taking like the same idea, but looking at it from an orthogonal point of view, right? Changing we didn't change sequence, nature changed sequence, but we used that to our advantage as a kind of a screen to try to answer the same question from a different perspective.
So how much of the motif needs to be changed? Is a SNP enough, or how much are we looking at?
Yeah, I mean, actually, we were kind of limited to SNPs because if you look at larger variants, it's like, so let's say you have an insertion, how do you even compare that to the other strain? Because it's a completely different system, right? And in that point, the sequence is so different. You don't really know how that affects things. So yeah, we limited ourselves to like 4 million SNPs between C57 Black 6 and BALB-C, and which, it didn't really affect that many sites. It was like maybe several hundreds for each of those factors.
Which should be enough, right?
Yes, and that turned out to be enough. Yeah, and so you wouldn't get, the effect was really, the strongest effect was so if you look at a transcription factor binding site, it's like a word, but some of these sites, some of the nucleotides in that motif, in the position weight matrix, they have more weight, right? Position weight matrix. So if the variant hits one of those heavily weighted, like major nucleotides that define most of the binding of the factor, then the effect is the strongest on binding of this factor as well as on the neighboring factor then indirectly. But if you have one of those more degenerate, like flanking sites or in between those strong sites, then the effect isn't as strong. It's more tolerant of variance there.
And then, go ahead.
The cool thing was, so we did this not just with the lineage determining factors that are, that seem to drive the open chromatin in the cell type that defines like how the cell is going to respond if you hit it with a signal, which is, then you bring in new factors that have to find their place in the genome, and they tend to go to the place that are already open, right? And so for those, their binding was often affected not by mutations, or by, you shouldn't call them mutations, really variants in their own motif, but in those motifs for those neighboring factors, right? And so there was an indirect also corroboration of our initial finding, So.
Yeah, in my intro, I said that you also were looking at epigenomics. So what did you do there? Looking at histone modifications, DNA methylation?
Yes. Yeah. So that was, we looked at mostly at histone methylation on histone H3 lysine 4. There's there's 3 different levels of methylation that you can get. And at the time, the monomethylation was like the enhancer mark. If you only have monomethylation but no trimethylation, and then trimethylation is pretty much mostly at promoters of genes. And I don't think it's really figured out yet why that is exactly. But it's linked at there's basically, you only find it if you have like mostly find it where there's a splice event happening next door. And so we looked at the one in between, dimethylation, between mono and tri, because that kind of marks both promoters and enhancers, and it's a little more localized than monomethylation. And at histone acetylation, in this case, acetylation 27. I think that's like one that just where the antibodies are the best. I mean, most acetylations are kind of correlated. So you can take any one, so why not take the one where your antibody works best? And then that's the one. So basically the effect of transcription factors, you could see then on the epigenetic level, because if you have transcription factor binding and you initiate transcription, regardless if it's at enhancers or promoters, you also get acetylation. And you could see that in the signal-dependent responses. And you can also see, we did this at nucleosome resolution with mRNA, so you could actually see the nucleosomes move apart when stuff would bind. So that was really cool to see,
So was this Nature publication already from your own group, or was this part of the postdoc project?
That was part of the postdoc project. I was a project scientist at the time.
Yeah.
And we had multiple related papers, that looked at, if you inhibit transcription in response to like a strong transcriptional stimulus like LPS and macrophages, then what happens to the epigenome, and where we could kind of deconvolute contributions of transcription factors and POL2 to the epigenetic readout that you get at the end.
Yeah, before we come to POL2, I wanted to ask another question, because you were a project scientist at UC San Diego, and then you moved to the Salk Institute as a director for next generation sequencing, and then you moved back to UC San Diego as assistant professor. So how did this come?
Yeah, that was serendipity, I think. So people at Salk, we had a collaboration with somebody in the Vermeer's lab, as well as at with Warren Evans and Rusty Gage and a bunch of different labs. And so they kind of knew me, and I had established a sequencing operation at this here in the glass lab, and was kind of involved. First sequencer in the core here and stuff. And so they were like, "Hey, we want to start new next gen sequencing core. Wouldn't you mind to be maybe the founding director?" And we have this big grant from the Helmsley Foundation in New York to get working on genomic medicine. That's the new focus. And I was like, wow, that sounds really cool. Everybody's they had like a plan on how to distribute the money and really make it work for everyone at the Salk, and to really start tackling genomic medicine aspects. And so, yeah, and then So I took that job. It was great. I love the SOC. It's really fun.
I mean, it's also a great location, right? Located at the sea, and the architecture is nice. So really great place.
Yeah, it's like a big family. It's kind of, I mean, you can have families with teenagers and stuff. You have the whole range of family in there, but it's a nice, it's a very cool place. Yeah.
So coming back to RNA Pol 2, you looked at how transcription elongation can affect the 3D structure of the genome. So how did you move into this direction and what did you find there?
Yes. So that was at SOC, actually. That started because I got involved in this big kind of, what was it, a U19 grant, I think, with an HIV and influenza researcher at SOC, and together with people in New York. And they had written this giant grant, or they were writing it actually, looking at, how does influenza virus infection affect the host genome and host transcription, and are there any druggable targets? Because at the time, this was long before COVID, right? Influenza was really one of the major killers in terms of viral infections across the globe. And so my colleague Chris, who was at that point the core director for bioinformatics at the SOC, and we wrote this pilot study on so influenza virus is an RNA virus, but unlike many other viruses, it doesn't replicate in the cytoplasm, but in the nucleus of the cells. And since we had been from, one of the aspects in the glass lab was also we were interested in how genome 3D structure affects transcription, and we couldn't really, that just never got published. And so we were like, you know what, we'll just do the same thing, but this time with influenza virus, and see if, does it affect the genome 3D structure, and how does that feedback on host transcription and host transcription regulation? So basically we started out doing just Hi-C, and there was just a new in situ Hi-C protocol that had come out that was much higher resolution than the previous versions that we had long worked on, and did this in cells that were infected at high MOI with influenza at different time points, and then looked at the genome 3D structure, and it was like, wow, the structure really. If you look at long time points, the structure just integrates. Like normally you have like chromosomal territories, and it's like chromosomes tend to interact with themselves mostly, and that those interactions, they kind of became weak, over like at 24 hours after infection, and then also more locally. Within chromosomes, there's topologically associating domains that were, discovered in Bing Wen's lab and Luke Decker's lab, and basically it's like the local domain structure. If you have the linear piece of DNA, it kind of crumples up into this kind of ball. At least that's the idea, right? And those, so you have boundaries, right? At basically where this kind of buds off from the linear genome, you have like these CTCF sites, which at the time was, let's see, was that known yet? I don't think that was known yet. Or it just came out then. Anyways, you have CTCF sites that are opposing, and they kind of cleave. No, they don't cleave off, but they, from there this domain holds. And so what we found was that with influenza, you get incredibly high antiviral gene transcription. The cell is trying to make some antiviral products to get rid of that thing that infected it. And at first, what we saw was that in some places, so looking at the large picture, the interactions between heterochromatic regions and heterochromatic regions, or euchromatin and euchromatin, which typically are a little separate, they would start to fade away, and you would have suddenly things in the heterochromatin making interactions with euchromatin where transcription occurs. So you have the silent chromatin that is typically stowed away, doing something with the active chromatin, which is like, how does this work, right? And then on a more local scale, we found that in different part of this big project, that there was transcription at the end of genes that normally wasn't there. Normally genes, have a beginning and an end, and that's it. And then pol 2, like it transcribes the gene, it falls off or it comes off and then restarts. But with influenza, it would just keep going. And those where it kept going, that was the places where suddenly new interactions would happen to places that this, if it ran into heterochromatin that wasn't transcribed, suddenly it would be transcribed and would suddenly make interactions with other euchromatic regions. It was crazy. It was like the whole structure of the genome got blown up and it seemed to be linked to transcription somehow. So that was very cool.
It's interesting and interesting also how the cell would recover from that.
Yeah. So it turned out that influenza A virus makes a non-structural protein 1 that interacts with the cleavage and polyadenylation machinery. So basically, you transcribe your mRNA, and then at some point, this together with pol 2, you have all this cleavage machinery traveling along, and it kind of scans the RNA. When it finds the polyadenylation site, it cleaves. And that cleavage leads to polyadenylation of the transcript that has just been made, as well as kind of loading of an exonuclease onto the remaining RNA that's coming out of the pol 2, which continues, and the idea is that this exonuclease kind of keeps chewing down until it hits the pol 2, and then there's some allosteric change or something that's not clearly defined yet, that leads for pol 2 to let go. But when this doesn't happen because the cleavage doesn't happen, this NS1 protein inhibits the cleavage itself. So pol 2 just keeps going. The transcript gets longer and longer and longer. And pol 2 never finds out that the gene's actually done. So this transcript was probably an antiviral transcript. And because it never got cleaved and polyadenylated, it doesn't get out of the nucleus. It stays in the nucleus. Then this means the antiviral protein doesn't get made. And the virus can just replicate to its heart's content. Meanwhile, the polymerase screws up the genome structure some more. And when we express this NS1 protein just from mRNA in the cells by electroporating mRNA, similar to the COVID vaccines, which was really useful back then to have as a technique, the cells would just die. They were not able to make any more protein because all of the stuff in the cells all the mRNA it would never become mRNA. The pre-mRNA would basically be stuck in the nucleus. And ultimately, after 24 hours this is highly overexpressed NS1 though, the cells would just not recover. They were all dead.
Interesting. So I don't know if this was exactly in that paper, but you also worked on an improved Chipstick protocol, which is also described in a STAR protocol. Chip has been around for a very long time, as you already alluded to. And methods like cut and take and cut and run are gaining popularity now. So I don't know exactly in which timely context this all was, like cut and take, cut and run, getting popular, you working on this Chipstick protocol. Yeah. But why did you think that working on an improved Chipstick protocol was necessary? And what did you do there?
That's a good question. So actually that Cell paper, it has one figure in it where we did like a time course, a Chipstick time course of pol 2, and you could actually watch pol 2 traverse a gene body, right? Like it was basically, and this only works when you have very strong stimulus where all the cells do the same thing and they start transcribing a gene that wasn't transcribed, right? So you can kind of watch it traverse, and that required like 72 Chipsticks. We did this for pol 2, CTCF and RAD21. It had to be very reproducible and it had to be fast. And so you can't do that with an old school Chipstick protocol where you have like one tube every time in phenylchloroform extraction or whatnot, right? And so I actually I have been working on chip protocols since 2002 when I was still in Regensburg, actually as a grad student. I just kept improving it over time. And so 2011 or something in the glass lab, I was like, we need a better Chipstick protocol because this sucks. I mean, like working, doing all these tubes and all this stuff. And so it was like, we got to miniaturize it, we simplify it, and we had a great tech. I think he's an MD now. He was amazing. Anyway, we said in 3 months, we kind of redid the Chipstick protocol, made everything on beats and optimized it. So the long-term goal was to make it, to be able to put it on a robot, like to really do this high throughput. And then one of the deciding things was that ChipExo had come out recently, and I tried to, so I applied that to there was for a different platform, though, I think iTorrent or something. And so I redid it for Illumina sequencing, which is different sequences stuff. And then as a control, I did like an experiment where I just did the regular Chipstick without, exonuclease digest of the DNA down to where the protein of interest is. And that totally worked. And I was like, wow, we can do everything in one tube, I think, right? And so, and then in 2017, I wanted to, use this protocol to try to kind of screen cancer cells, like, for patient samples from thousands of patients. There's a tissue bank here that has lots of samples available. And so we further optimized that. And without that protocol, we wouldn't have been able to do that cell paper because so many Chipsticks. And so I had a great postdoc. She's in Switzerland now, and she did she optimized that protocol and did hundreds of Chipsticks just to optimize it. And so that helped us both, speed it up and scale it down because, with influenza infected cells, you don't get a lot. And so we had to be able to not use like 10 million cells, but maybe like 100,000, and with this really helped, pulling that project off because these were all primary cells, right? At least the ones that we got from New York. And, and the only thing that was missing was like we still had to do probe sonication because all the sonicators we tried, they didn't really work so great. So it was like we tried like everything under the sun.
Yeah, I mean it's still primary cells, right? So
sorry,
it's still primary cells, and they are kind of tricky in the end.
Yeah. No, it was just more the reproducibility of most sonicators out there. Like anything that wasn't more than a single thing. You'd always have some sort of weird dropouts, and there was tricks to kind of work around it. But it's like, if you do get primary cells, you'd want to make sure that part works and not like, find out later, like, actually, sorry, that one sample just didn't work. I don't know why, Yeah. Anyway, so yeah, that was a major breakthrough for the lab, just to be able to do this in a higher throughput and very reproducibly.
So you're still using this approach nowadays?
Yes. Yeah. So, because you can use it for anything. I mean, we've done Chipstick in human retinas, for example, or sorted mouse cardiomyocytes where you just don't get a lot, or mouse pituitaries, like 60,000 cells and stuff. You can't sample, but you can still do Chipstick. It's pretty cool.
Interesting. And this feeds directly into my next question, because this is the beginning of the end, basically. And I've thought of a really great question, but it seems that at least the first part of this question is not true for most of them, because as you told me in the beginning, you are currently in the phase of grant writing, so you don't have to imagine that, but you are due to submitting a grant proposal soon. So what do you write into those grant proposals?
What do I write into? So what am I am I into now? Right?
Exactly.
Yeah. So over the last, through COVID, I think we kind of established some interesting, massively parallel reporter assay variants that so basically the chromatin stuff was kind of dormant for a while. We did like try some cut and run, or I still want to try cut and tag, but I don't know. I've seen kind of weird data artifacts there where I'm not sure, and I don't really have time to figure that out for myself. So I'll probably stick with what I know, which is Chipstick and High-C and stuff like that. So we've been working on similar to our first and the second paper. The first one was about looking where do the proteins go, and the second one was what happens if I change the sequence, right? So that part is, you can do this in a massively parallel way where you take synthetic sequences, put them in a reporter, which is the first time enhancers were found, in basically a plasmid where promoter transcription was incredibly activated because there was this extraneous piece of DNA in there. And you can do this with simultaneously with thousands, tens of thousands of sequences. And we've modified it. We later found out other people had already modified it that way, but we've modified the protocol where instead of having the barcode at the end of a reporter G, we put it at the very beginning. Each sequence gets a barcode, and that helps you tell, by just counting the barcodes, you can tell what the sequence was upstream that drove that transcription. And so we did this initially for promoters, and now we're working on expanding this into, looking at, so you can do this at base resolution. You can look at transcription initiation and how it gets affected by upstream sequences, which, it turns out that most of these things have been done either indirectly, where like if you don't look at transcription initiation yourself, you just get a level, you can do saturation mutagenesis, but you can't really, because you can't measure where the initiation happens in an old school reporter assay, you don't actually know what happens. You can just look at the levels. And if the pattern of initiation changes, you can't actually test that directly. So with this assay, you can, and then we're expanding this into enhancer assays. And then I want to actually do more work in terms of using this for more advanced things where we can interrogate what binds there. That hasn't really been done at all. So that's kind of what I'm really interested in now. And then use this in some sort of differentiation system. So the thing with enhancers is, when we study them by an MPRA, we typically take a cell and we stick the enhancer in there and say, oh, look, it works, or it doesn't, at least in the mammalian system. But what, for example, people in Drosophila or C. elegans have seen a long time ago is that, these things are active during differentiation. But if you have a cell that doesn't differentiate, you don't actually know what it does. Like you have the transcriptional effect directly, but you're not looking at the trajectory of what happened during, the differentiation and what did the sequence actually do, right? So that's where we're kind of pivoting into pluripotent stem cells and then using them as a differentiation system where we can kind of combine these two things. And that's what I'm most excited about right now.
Mm-hmm.
Still a bulk thing. It's not single cell or whatever, what most people are doing, but most of those transcriptional things you can't do them in single cells. It's kind of, I wish I could.
So for the last 37 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I'd like to add something. So career-wise, you can tell that I've been bouncing around. Like I'm a trained chemist. I kind of got into biochemistry, but in medicine. I'm actually a professor in medicine now. I think, I don't know. This was not a planned event. Not this. I think I don't know. I feel like I only got here because I kind of stuck with what interested me, like many people do. Or there's a tendency, for myself too. There's lots of self-doubt. Am I doing the right thing? Where is this going? Also, scientific questions. Where is this going? Especially now, where you have hundreds to thousands of preprints and papers coming out. What is relevant? And I feel that just sticking to your own compass is kind of the best way to, like what am I interested in? That helps kind of prioritize things and somehow got me to where I am now, and also scientifically, I feel that I used to be very doubtful of what I was doing. And then it's like, oh, but this paper, like our Malt cell paper got at this point got cited probably 10,000 times or something, right? That wasn't the goal.
Yeah.
It was like, oh, this is really cool. I don't understand how it works, But if you have things that you, especially where you don't know and you kind of have a hunch, and sometimes it feels dangerous to follow that up. But if you don't, then what are you doing? Right?
Mm-hmm. Yeah, you were not confident of your work in the first place.
Yeah, in the beginning I wasn't, but now I'm kind of more confident in pursuing things that are not super mainstream, just because it's worked in the past, and I don't know. So it's a balance. The most important thing is, I guess, to talk to people, like talking to you or talking to my colleagues and stuff. Like, does this make sense? Is this going the right way? I think that with COVID, it became really obvious that how important it is to talk to people in science.
Yeah,
when you can't, because nobody's here.
Yeah, I think that's a good point to end this interview. So thank you, Sven, for your time and for being on the show.
Yeah, it was a pleasure. Thanks for having me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.